Hello there, and welcome to Uniscope, where we let curiosity slow down to the pace of geological time and wonder takes its time to unfold. Tonight, we're not looking up at the stars or forward into the future. Instead, we're stepping backward, far backward, to a version of Earth that would feel like walking through an alien landscape, yet one that's quietly familiar in ways that might surprise you. We're going to visit a time when the tallest living things on our planet weren't redwoods or oaks, or even the earliest primitive trees. They were mushrooms, massive, towering fungi that stood like ancient pillars across continents that were still finding their shape. A world where spores, not pollen, filled the air, and where the largest structures on land were built not of wood, but of something far stranger and more mysterious. If that sounds like something from science fiction, well, it's not. It's science fact. And it happened right here, on this same Earth we call home, about 400 million years ago, during a chapter of our planet's story called the Devonian period, a time when the very concept of a forest meant something completely different than it does today. Before we begin this quiet journey into the deep past, I'd love to know where you're listening from tonight and what time it is in your part of the world. It's strange to think that no matter when or where you are, we're both inhabitants of the same planet that once belonged to these fungal giants. If this kind of gentle exploration into Earth's hidden history is something you enjoy, I'd be grateful if you considered tapping like or subscribe. It helps this small signal reach other curious minds who might enjoy wandering through time with us. So settle in wherever you are. Let your breathing slow down to match the patience of deep time itself. We're about to explore a world that existed for millions of years before the first tree ever reached toward the sky. A world where Earth was experimenting with life in ways that seem almost unimaginable now. When we think about the history of life on Earth, we often picture a steady progression. First the oceans filled with life, then creatures crawled onto land, then forests appeared, then animals grew larger and more complex. But the real story is messier, stranger, full of experiments that didn't last and innovations that seem almost accidental. Life doesn't follow a script, it improvises, constantly trying new approaches, new solutions, new ways of being alive. The story of life is more like jazz than classical music, more like a conversation than a monologue. Each organism that has ever lived has been both responding to the world it inherited and creating the world that will come after it. And sometimes those experiments take directions that seem impossible to predict, that challenge everything we think we know about how life works. 400 million years ago, during the Devonian period, Earth was in the middle of one of these great experiments. Life had been exploring the possibilities of land for about 100 million years by then, but it was still figuring out the basic rules. How do you grow tall without falling over? How do you transport water from soil to sky? How do you compete for sunlight when there's no playbook for what a forest should look like? How do you even define success when you're the first of your kind to try something entirely new? These weren't just technical problems, they were existential ones. Every organism that ventured onto land was essentially asking the question, what does it mean to be alive in this new medium? Water had been the cradle of life for over 3 billion years. It provided support, it carried nutrients, it protected organisms from the harsh radiation of the sun. But land offered something that water couldn't: the possibility of height, of reaching toward the sky, of accessing the unlimited energy of direct sunlight. At this time, our planet would have been almost unrecognizable to modern eyes. The continents were arranged in a completely different configuration, clustered together in ways that seem strange when we map them onto our mental image of the world. There was no Atlantic Ocean as we know it. Instead, most of the land masses were gathered together in the southern hemisphere, forming a massive supercontinent called Gondwana, while smaller continents drifted in warm, shallow seas that covered much of what would eventually become North America and Europe. The geography of the Devonian world was dominated by vast, shallow seas and enormous river deltas. Much of what we now call North America was covered by the Kaskaskia Sea, a warm, clear ocean that stretched from what is now the Gulf of Mexico to the Arctic Ocean. Europe was an archipelago of tropical islands. Asia was fragmented into several smaller continents. The familiar outlines of our modern world were nowhere to be seen. But perhaps most alien of all was the climate. The Devonian was what geologists call a greenhouse period, a time when Earth's climate was driven by high levels of carbon dioxide in the atmosphere. There were no ice caps, no frozen poles, no seasons as we understand them today. The temperature difference between the equator and the poles was much smaller than it is today. Tropical conditions extended almost to what we would now consider the Arctic regions. The air was thick with moisture, and carbon dioxide levels were perhaps 3 to 5 times higher than today, creating a greenhouse effect that kept temperatures stable across the globe. There were no deserts as we know them, no tundra, no snow-covered mountains. Instead, Earth was wrapped in a blanket of warmth and humidity that extended from what we now call the tropics all the way to the poles. It was a world where ice was essentially unknown, where winter meant perhaps a slight cooling and a change in rainfall patterns, but never the dramatic seasonal transformations that define so much of modern life. And in this warm, wet world, something remarkable was happening. Across the landscape, strange towers were rising, not made of stone or wood, but of fungal tissue. Structures that could grow as tall as a 2-story building and as wide as a small room. They had no leaves, no branches, no flowers, just smooth, pale columns reaching upward, some branching at the top like massive candelabras, others standing solitary and straight like biological lighthouses in a landscape that was still learning what it meant to reach toward the sky. These were called prototaxites, and for the longest time, scientists weren't even sure what they were. When the first fossils were discovered in the 1800s, researchers found themselves looking at something that didn't fit any known category. The fossils were found in deposits around the world, from Scotland to North America to Australia, suggesting that whatever these organisms were, they had achieved a global distribution that spoke to remarkable evolutionary success. Some thought they might be early trees, perhaps relatives of conifers that had somehow grown to enormous size. The sheer scale of the remains suggested something that had mastered the art of growing tall, and trees seemed like the most obvious explanation. Others suggested they were giant algae, massive versions of the seaweeds that flourished in the oceans. The organic composition of the fossils seemed consistent with algal tissue, and the idea of marine organisms adapting to terrestrial life wasn't unprecedented. Still others proposed they might be some kind of primitive plant that we simply hadn't seen before, something that represented an entirely different approach to the problem of being a large terrestrial organism. The Devonian was a time of incredible botanical innovation, when plants were experimenting with solutions to problems that had never been solved before. Perhaps prototaxites represented one of these experiments, a path not taken in the evolution of large land plants. The fossils themselves were puzzling in their simplicity. Unlike tree fossils, which show clear evidence of growth rings, complex branching patterns, and the intricate vascular systems that plants use to transport water and nutrients, prototaxites' remains were strangely uniform. They showed a honeycomb-like internal pattern, with tubes and chambers running through the structure, but no clear organization that suggested leaves, roots, or the complex tissue differentiation that characterizes modern plants. The surface texture was unlike anything familiar. Not the complex bark patterns of trees, not the smooth surfaces of succulent plants, not the delicate structures of ferns or mosses. Instead, the fossils suggested something with a waxy, almost translucent quality, something that seemed to glow from within when the light hit it just right. This wasn't the robust woody tissue of trees or the delicate cellular structure of typical plants. It was something else entirely. It wasn't until much more recently, with advances in microscopy and chemical analysis, that we began to understand their true nature. Researchers began to examine the cellular structure of the fossils in unprecedented detail, looking at the arrangement of individual cells, the composition of cell walls, the patterns of tissue organization. They analyzed the chemical signatures preserved in the fossils, looking for clues about the metabolism and biochemistry of these mysterious organisms. The internal structure, the chemical composition, the way they grew, all pointed to a single conclusion that seemed almost impossible to believe. They were fungi, the largest living things on land at the time, and possibly the largest fungi that have ever existed. Not mushrooms as we think of them today, with their familiar caps and stems, but something far more ambitious, far more monumental. Something that represented an entirely different approach to being a fungus, an approach that involved reaching toward the sky with the same ambition that would later characterize trees. Imagine walking through this landscape. Picture yourself stepping carefully across the spongy ground, covered in a thick mat of primitive mosses and lichens that squelch slightly under your feet, releasing the rich, earthy smell of decomposing organic matter mixed with the fresh scent of growing plants. The ground itself would feel different under your feet, less solid than modern soil, more like walking across a vast living carpet that gives slightly with each step. The air around you is warm and heavy, almost like breathing in a greenhouse, thick with humidity that seems to cling to your skin. Each breath carries the scent of moisture and growth, of spores and decay, of a world where the boundary between life and death is blurred by the constant cycle of decomposition and renewal. The humidity is so thick you can almost see it, creating a soft haze that filters the sunlight and gives everything a dreamlike quality. Small arthropods, early relatives of insects and spiders, scuttle through the undergrowth, their movements creating tiny rustling sounds in the primitive vegetation that carpets the ground. These creatures are unlike anything you might see today. Experimental forms that are still working out the basic problems of living on land. Some have too many legs, others too few. Some have simple eyes, others seem to navigate by touch and chemical sensation alone. They represent life's early attempts to colonize the terrestrial environment, each species a rough draft in evolution's ongoing experiment with land-based existence. The arthropods of the Devonian were pioneers in their own right, facing many of the same challenges as the plants around them. How do you support your weight without the buoyancy of water? How do you keep from drying out in the open air? How do you find food and mates in a three-dimensional environment where scent trails dissipate quickly and visual landmarks are few? These small creatures were solving these problems through trial and error, developing solutions that would eventually lead to the incredible diversity of terrestrial arthropods we see today. Some of these early arthropods were quite large by modern standards. There were millipede-like creatures that could grow to over a foot in length, their segmented bodies undulating through the undergrowth like terrestrial snakes. There were early arachnids, some as large as dinner plates, hunting smaller prey through the tangled vegetation. And there were the first primitive insects, still clumsy flyers that spent most of their time crawling through the leaf litter, but occasionally taking to the air in short, ungainly flights. The air itself would be different, not just in temperature and humidity, but in its very composition. Each breath would contain more carbon dioxide than you're used to, giving everything a slightly different quality, a richness that might make you feel slightly lightheaded if you were somehow transported there from our modern world. The high carbon dioxide levels would also affect the way plants grew, allowing them to photosynthesize more efficiently, but also creating an atmosphere that would feel alien to modern lungs. And floating through this thick air would be clouds of spores instead of pollen, millions upon millions of fungal reproductive cells drifting on every breeze, settling on every surface, turning the air itself into a living medium. The spores would be everywhere, coating leaves with a fine powder, creating visible streams in the slanted rays of sunlight, accumulating in the still corners where air currents converged. Unlike the seasonal pollen releases we're familiar with today, spore production in the Devonian world was more constant, more pervasive, creating an atmosphere that was always thick with the potential for new life. These spores represented hope and possibility in their most concentrated form. Each one carried the complete genetic blueprint for a new organism, the potential to establish a new colony in some distant location, to contribute to the slow but inexorable spread of life across the terrestrial landscape. They were messages in bottles, cast into the aerial ocean by organisms that had never seen where their offspring might land. And all around you, rising from this alien ground, these pale towers would stand. Some solitary, like ancient obelisks marking unknown graves, their surfaces smooth and unmarked by time. Others clustered in groups, creating grove-like formations that cast strange shadows across the landscape, their arrangement suggesting some organizing principle that we can only guess at. Their surfaces would be smooth, almost waxy, with a texture unlike anything in our modern world. Not the rough bark of trees, not the smooth skin of succulent plants, but something in between, something that speaks of an entirely different approach to being a large living structure. The colour of these towers would have been unusual as well. Not the familiar browns and greys of tree bark, not the vivid greens of chlorophyll-rich plants, but something more subtle, more mysterious. Pale yellows and creams, perhaps with hints of pink or orange where the internal tissue showed through the translucent outer layers. In certain lights, they might have seemed to glow from within, their internal structure creating patterns of light and shadow that shifted as the sun moved across the sky. The surfaces of the towers would have been marked by subtle patterns, the external evidence of the complex internal structure that supported these massive organisms. Gentle ridges and depressions arranged in spiraling patterns that followed the growth of the organism, creating a natural architecture that was both alien and oddly beautiful. These patterns would have been most visible near the base of each tower, where the fungal tissue was oldest and most developed, becoming more subtle toward the growing tips where new tissue was still being laid down. There would be an eerie quiet to this world that would strike you immediately. No bird songs, because birds wouldn't exist for another 150 million years. No rustling of leaves in the wind, because true leaves were still rare, and those that existed were small, simple affairs that hugged the ground. No buzzing of bees or chirping of crickets, because the complex relationships between insects and flowering plants were still inconceivably far in the future. Instead, the soundscape of the Devonian would have been dominated by subtler sounds. The constant dripping of water from the humid air, condensing on every surface and running down the sides of the fungal towers in tiny rivulets. The gentle creaking and settling of the towers themselves as they swayed in the wind, their fungal tissue flexing and adjusting to the constant movement of air around them. The soft rustling of small arthropods moving through the undergrowth, their chitinous legs clicking against stones and primitive plant stems. Wind moving through and around the fungal structures would have created sounds unlike anything we hear today. The smooth surfaces and unusual internal structure of the towers would have acted like enormous wind instruments, creating low, almost musical humming sounds as air passed around and through them. The frequency of these sounds would have varied with wind speed and direction, creating an ever-changing acoustic environment that served as a kind of aerial symphony. The voice of the wind made audible through fungal architecture. The scale of these fungal towers is difficult to grasp without seeing them yourself. The largest prototaxites specimens we've found in the fossil record suggest structures that could reach 24 feet in height and 3 feet in diameter. To put that in perspective, that's taller than most modern telephone poles, wider than most tree trunks you'd encounter in a typical forest, built entirely of fungal tissue. Standing next to one of these giants, you would have felt like a small child looking up at an enormous monument, something built on a scale that dwarfed human experience. But the height alone doesn't capture the full impressiveness of these structures. The volume of living tissue represented by a single large prototaxites tower was enormous, perhaps equivalent to several modern trees combined, and this tissue wasn't the relatively simple, largely dead wood that makes up most of a tree's bulk. It was living fungal tissue, metabolically active, constantly growing and changing, requiring a constant supply of nutrients from the soil and producing the enormous quantities of spores that kept the reproductive cycle going. They didn't grow quickly like the mushrooms you might find sprouting in your garden after a rain, those familiar fungi that seem to appear overnight and disappear just as quickly. Instead, they built themselves slowly, patiently, layer by layer, season by season, perhaps taking decades or even centuries to reach their full impressive height. This slow growth was part of what made them so successful. They weren't rushing toward reproduction like modern mushrooms. They were building monuments to persistence, structures that could stand for centuries and serve as spore-producing platforms for generation after generation. The growth pattern of these giants would have been fascinating to observe if you could have watched it over the appropriate timescale. New tissue would have been added primarily at the growing tips, with the base of each tower remaining relatively stable once it had been established. This meant that the oldest parts of each structure were at the bottom, while the youngest, most active growth was happening at the top, sometimes hundreds of feet above the forest floor. As they grew, the towers would have developed their characteristic branching patterns, with new growth points splitting off from the main stem to create the candelabra-like structures that we see in the fossil record. These branches weren't random. They followed specific developmental programs, genetic instructions that had been refined over millions of years of evolution to optimize spore dispersal and structural stability. But how did they manage to grow so large? This question puzzled scientists for years after we first understood what prototaxites actually was. Modern fungi are mostly decomposers, organisms that specialize in breaking down dead organic matter and recycling nutrients back into ecosystems. They tend to be relatively small, hidden, working their magic beneath the surface or in the shadows of larger plants. They're the janitors of the natural world, essential but rarely spectacular. What allowed these ancient fungi to become the giants of their time, the skyscrapers of the Devonian landscape? The answer lies partly in the unique nature of the Devonian world itself. This was a time when the boundary between land and water was still blurry, still being negotiated by life itself. Much of what we might generously call land was actually more like vast wetlands, endless swamps and tidal flats that disappeared and reappeared with the rhythms of ancient tides. The distinction between aquatic and terrestrial environments was much less clear than it is today. These wetland environments were perfect for fungi. They provided the constant moisture that fungal tissue requires, while also accumulating vast quantities of organic matter from the primitive plants and algae that lived and died in the shallow waters. The soil, such as it was thin and poor in the traditional nutrients that plants need, but it was rich in the organic compounds that fungi specialize in breaking down and utilizing. There weren't many large plants competing for space or resources in these early terrestrial environments. The first true trees were still millions of years in the future. The plants that did exist were small, primitive affairs, mostly mosses and early ferns that rarely grew more than a few feet tall. This meant that there were vast areas of landscape where nothing was competing for the vertical space that the fungal towers were beginning to occupy. Unlike plants, fungi don't need to photosynthesize. They don't need to capture sunlight directly, don't need to worry about positioning their tissues for maximum light exposure, don't need to compete for the best spots in the sun. Instead, they can feed on organic matter in the soil, breaking down the remains of smaller plants and microorganisms, recycling death into life through chemical processes that are both elegant and efficient. This gave them a fundamental advantage in the early terrestrial environment, while plants were still struggling to solve the basic engineering problems of terrestrial life: how to transport water from soil to sky, how to support their own weight without the buoyancy of water, how to prevent desiccation in the open air. Fungi could sidestep many of these challenges by focusing on decomposition rather than photosynthesis. In a world where growing tall was a challenge for photosynthetic organisms, where the engineering problems of building a tree had not yet been solved, fungi found a different strategy altogether. They could build their towers without worrying about leaves or light capture. They didn't need to solve the complex problems of how to transport water hundreds of feet up from roots to crown, how to maintain structural integrity in the face of wind and weather, how to balance the competing demands of height and stability. They just needed to access nutrients in the soil and disperse their spores as widely as possible. And for that, height was everything. The taller a fungal tower could grow, the farther its spores could travel, the larger the area it could potentially colonize with its offspring. In the flat, low-lying landscapes of the Devonian world, even a modest increase in height could dramatically increase the range of spore dispersal. And disperse they did. The top of a Prototaxites tower was essentially a massive spore-producing factory, a biological launch pad designed to send millions of reproductive cells into the atmosphere with each reproductive cycle. The reproductive structures at the tips of the towers were unlike anything we see in modern fungi. Instead of the familiar mushroom caps that we associate with fungal reproduction, these ancient giants produced their spores through specialized structures that we can only partially reconstruct from the fossil record. By growing tall, these fungi could release their spores into the wind currents that flowed across the Devonian landscape, spreading them across vast distances with an efficiency that would make any modern distribution network envious. The physics of spore dispersal meant that even a small increase in release height could result in a dramatic increase in dispersal range. Spores released from a height of 24 feet could potentially travel hundreds of times farther than those released from ground level. In a world with few geographical barriers and consistent wind patterns created by the uniform warm climate, a successful spore could potentially travel hundreds or even thousands of miles before settling and beginning to grow. This meant that a single successful Prototaxites tower could potentially seed new colonies across entire continents, creating a network of fungal outposts that spanned the known world. The global distribution of Prototaxites fossils suggests that this strategy was remarkably successful. We find their remains on every continent, rock formations that span millions of years of geological time. This widespread distribution speaks to an organism that had mastered the art of long-distance dispersal, that had found a way to colonize virtually every suitable habitat on the planet. The internal structure of these fungal giants was unlike anything we see today, and examining fossilized cross-sections reveals an intricate architecture that speaks to millions of years of evolutionary experimentation. Instead of the solid wood of trees or the simple cellular structure of modern mushrooms, prototaxites shows a complex pattern of tubes and chambers, almost like a honeycomb, or the internal structure of bone. These passages likely served multiple purposes, transporting nutrients from the base to the growing tip, providing structural support that allowed the organism to reach such impressive heights, and perhaps even allowing for air circulation within the massive structure. The tube system was remarkably sophisticated, with different sized passages arranged in complex patterns that suggest a high degree of internal organization. The largest tubes, some nearly an inch in diameter, probably served as the main highways for nutrient transport, carrying dissolved minerals and organic compounds from the base of the tower to the growing tips. Smaller tubes, branching off from the main channels, would have distributed these nutrients to the living tissues throughout the structure. The smallest passages, barely visible even under magnification, might have served for gas exchange, allowing oxygen and carbon dioxide to move through the tissue. Some researchers have suggested that prototaxites might not have been a single organism at all, but rather a complex community of different fungal species living together in a kind of biological apartment building. Different zones within the structure might have been occupied by different species, each contributing to the overall growth and stability of the tower. Some might have specialized in nutrient absorption from the soil, others in structural support, still others in spore production and dispersal. If this interpretation is correct, then these ancient giants were early experiments in cooperation, biological cities rising from the primordial landscape, proof that even in the earliest days of complex life on land, organisms were already discovering the power of working together. The idea of multi-species fungal communities isn't unprecedented in the modern world. Many of the largest fungal organisms alive today are actually complex networks of different species that have learned to cooperate for mutual benefit. The metabolic processes that powered these giants would have been fundamentally different from those of modern plants. Instead of using photosynthesis to capture energy from sunlight, they relied on the chemical breakdown of organic matter. This process, called chemosynthesis, involves breaking down complex organic molecules and extracting energy from the chemical bonds that hold them together. This gave the fungal towers access to energy sources that were unavailable to photosynthetic organisms. They could tap into the vast reserves of organic matter that accumulated in the Devonian wetlands, breaking down everything from primitive plant matter to the remains of early animals. They were the recyclers of the Devonian world, taking the waste products of other organisms and turning them into the raw materials for their own growth. The scale of this recycling operation would have been enormous. A single large prototaxites tower might have processed tons of organic matter during its lifetime, breaking down everything within reach of its extensive underground networks and converting it into new fungal tissue. They were living composting systems, biological recycling plants that turned death into life on an industrial scale. The world of the mushroom rulers wasn't just about the giants, though. The Devonian period was a time of incredible innovation across the entire fungal kingdom. Smaller fungi were developing new ways of living, new partnerships with the primitive plants that were beginning to evolve, new strategies for survival in a world that was constantly changing. This was when the first complex relationships between plant roots and fungi, called mycorrhizae, began to develop and flourish. These partnerships represented one of the most important innovations in the history of life on Earth, a collaboration that would become so fundamental to terrestrial ecosystems that most land plants today couldn't survive without their fungal partners. The development of mycorrhizal relationships wasn't a sudden event, but rather a gradual process of coevolution that took place over millions of years. Early plants struggling to extract nutrients from the poor soils of the Devonian world gradually learned to work with soil fungi that were better equipped to access mineral resources. The fungi, in turn, gained access to the sugars that plants produced through photosynthesis. These partnerships were revolutionary in their implications. The fungi could extend the reach of plant roots, sending their thread-like hyphae far into the soil to access water and nutrients from a much larger area than the plant could reach on its own. They could break down organic matter that the plants couldn't process, extract minerals from rock that would otherwise be unavailable, and even help plants communicate with each other through chemical signals passed through the fungal network. The fungal networks that formed during the Devonians were the ancestors of the complex underground webs that connect modern forests. Even in these early stages, they were beginning to demonstrate the kinds of emergent properties that would later make them so important to ecosystem function. Plants connected to the same fungal network could share resources, with healthy individuals supporting their struggling neighbours through the underground connections. In return, the plants shared some of the sugars they produced through photosynthesis, providing the fungi with a steady source of energy that didn't depend on finding dead organic matter to decompose. It was a trade deal that would become one of the most important relationships in the history of life on Earth. A partnership so fundamental that most land plants today couldn't survive without their fungal allies. But in the Devonian, these partnerships were new, experimental, still being worked out through trial and error. The early plants were small, tentative things. Primitive ferns unfurling their fronds close to the ground, early mosses creating carpets of green in the wettest areas, and the very first woody plants, the ancestors of all modern trees, just beginning to appear as small shrubs that rarely grew more than a few feet tall. These early plants were pioneers in their own right, facing challenges that no organism had ever faced before. They were learning how to extract water from soil instead of simply absorbing it from their surroundings. They were developing new ways to support their own weight without the buoyancy of water. They were experimenting with different strategies for preventing water loss in the open air. The first primitive ferns were perhaps the most successful of these early land plants. They had developed relatively sophisticated vascular systems for transporting water and nutrients, and their characteristic fronds were efficient at capturing sunlight for photosynthesis. But even the largest of these early ferns were small by modern standards, rarely growing more than a few feet tall, creating low carpets of green that covered the wettest areas of the Devonian landscape. Early mosses represented a different approach to terrestrial life. Instead of developing complex vascular systems, they remained relatively simple, absorbing water and nutrients directly through their surfaces. This strategy worked well in the humid Devonian climate, but it also limited their size and restricted them to the wettest microhabitats. The first woody plants were just beginning to appear during this time, tiny shrubs that represented the earliest experiments in building woody tissue for structural support. These were the distant ancestors of all modern trees, but they were still crude prototypes, barely more than enlarged moss plants with slightly more sophisticated water transport systems. These early plants were still learning how to be plants. They were figuring out how to support their own weight, how to transport water efficiently, how to make the most of the sunlight that filtered down through the humid atmosphere. They were solving problems that had never been solved before, developing solutions that would eventually lead to the complex plant communities we see today. Against this backdrop of small, cautious plant life, the fungal towers must have seemed like giants from another world, monuments to a completely different approach to the problem of being alive on land. While the plants struggled with the basic challenges of terrestrial existence, the fungi had found a way to bypass many of these problems by focusing on decomposition rather than photosynthesis. The contrast between the small, struggling plants and the towering fungi would have been dramatic. In a landscape where most organisms hugged the ground, rarely reaching more than a few feet in height, the Prototaxites towers stood like biological skyscrapers, dominating the visual landscape in a way that nothing else could match. The air in this ancient landscape would have been different in ways that go beyond temperature and humidity. The very composition of the atmosphere was alien by our standards. Oxygen levels were still rising slowly, approaching modern levels but not quite there yet, hovering around 15 to 16% compared to today's 21%. This might not sound like a significant difference, but it would have been noticeable, making the air feel slightly thinner, perhaps leaving you feeling as though you were at a moderate altitude even at sea level. The lower oxygen levels would have affected not just respiration but also combustion. Fires would have been less intense and less common than in our modern world. This had important implications for the evolution of terrestrial ecosystems, as fire plays a crucial role in many modern plant communities. The Devonian world was largely a fire-free environment, which contributed to the accumulation of organic matter that provided so much food for the fungal decomposers. Carbon dioxide was much more abundant, perhaps 3 to 5 times more concentrated than in our modern atmosphere. This created the greenhouse effect that kept the planet warm from pole to pole, but it would also have given the air a different quality, a richness that might have made you feel slightly drowsy if you were suddenly transported there from our modern world. The high carbon dioxide levels also meant that photosynthesis was more efficient, allowing plants to grow faster with less water stress than they would experience in our modern atmosphere. And floating through this thick, carbon-rich air were clouds of fungal spores, billions upon billions of reproductive cells drifting on every breeze like an endless invisible snowfall. The concentration of spores in the Devonian atmosphere would have been orders of magnitude higher than anything we experience today. Every breath would have carried thousands of fungal reproductive cells. Every surface would have been constantly colonized by new arrivals carried on the wind. These spores were the messengers of the fungal kingdom, carrying genetic information across continents, seeding new colonies in distant lands, connecting far-flung populations through the simple act of reproduction. Unlike the heavy seeds that plants would eventually evolve, spores were light, almost weightless, designed to travel as far as possible on the slightest air current. The physics of spore dispersal is fascinating and would have been crucial to the success of the Devonian fungi. Spores are essentially single cells surrounded by a protective wall that can resist desiccation, temperature extremes, and other environmental stresses. They can survive incredible journeys, lying dormant for months or even years until they find exactly the right conditions to germinate and begin growing. The shape and surface texture of spores are carefully evolved to optimize their aerodynamic properties. Many fungal spores are perfectly spherical, the ideal shape for remaining suspended in air currents. Others have specialized surface features, tiny ridges or projections that create turbulence and help keep them aloft. Some even have tail-like appendages that help them spin as they fall, slowing their descent and increasing their dispersal range. In the Devonian world, spore dispersal was one of the most important processes shaping the landscape. The tall prototaxite towers were essentially massive spore-launching platforms, biological radio towers broadcasting genetic information across the ancient world. With each reproductive cycle, they would release millions of these tiny time capsules into the atmosphere, each one carrying the potential to establish a new colony in some distant location. The timing of spore release would have been carefully coordinated with weather patterns and seasonal cycles. Even in the relatively stable Devonian climate, there would have been optimal times for dispersal, periods when wind patterns and humidity levels created the best conditions for long-distance transport. The fungi would have evolved internal biological clocks that synchronize spore production with these favourable periods, but not every spore would find a suitable home. The vast majority would fall in places where conditions weren't quite right, where the humidity was too low, or the nutrients were insufficient, or the local ecosystem was already too crowded with competitors. They would lie dormant in the soil, waiting for conditions to change, waiting for their moment. Some would wait forever, becoming part of the geological record, tiny fossils that we can find today, embedded in rocks from the Devonian period. The successful spores, the lucky ones that found perfect conditions, would begin the slow process of growth that could eventually lead to another towering giant. But this process was neither quick nor guaranteed. A spore might germinate and begin growing, only to be eaten by one of the small arthropods that crawled through the undergrowth. It might establish itself successfully, only to be washed away by flooding or buried by sediment. Even those that survived these early challenges faced the long, slow task of growing large enough to become a significant presence in the landscape. The transition from a newly germinated spore to a mature reproducing tower might take decades or even centuries. This meant that successful colonization of new areas was a multi-generational process, requiring not just the initial establishment of a colony, but also its long-term survival and eventual reproduction. The growth patterns of young prototaxites would have been quite different from those of mature towers. Newly established colonies would have started as small, inconspicuous patches of fungal tissue, spreading outward through the soil as they accumulated nutrients and biomass. Only after reaching a certain critical size would they have begun the transition to vertical growth, slowly building the towers that would eventually dominate the landscape. This two-phase growth pattern, horizontal expansion followed by vertical construction, would have been crucial to the success of these organisms. The initial horizontal phase allowed them to establish a broad base of nutrient extraction, creating the foundation that could support the enormous biomass of a mature tower. The vertical phase then allowed them to access the aerial dispersal opportunities that made long-range colonization possible. As we try to picture this world more clearly, it's worth spending time thinking about the daily rhythms of life in the age of mushroom rulers. Without the familiar cycles of deciduous trees, without the seasonal migrations of large animals, without even the daily chorus of birds, the passage of time would have been marked by different phenomena entirely. The days themselves were shorter than ours. The Earth was spinning faster 400 million years ago, completing a full rotation in about 22 hours instead of our current 24. This meant that the sun would rise and set more quickly, creating a rhythm of light and shadow that would feel slightly accelerated to our modern senses. The shorter days also meant that there were more days in a year, about 400 compared to our current 365. But with the thick, humid atmosphere and the generally overcast conditions that prevailed across much of the planet, the distinction between day and night would often have been subtle, more of a gradual brightening and dimming than the sharp transitions we're used to. The high humidity would have created frequent clouds and fog, filtering the sunlight and creating a soft, diffused lighting that gave the landscape an ethereal, dreamlike quality. The seasons, if they existed at all, would have been gentler than anything we experience today. The warm, stable climate meant that temperature variations were minimal. There was no dramatic autumn leaf change, no winter dormancy, no spring awakening. Instead of the sharp, seasonal transitions that drive so much of modern ecology, the rhythms of life in the Devonian were probably driven more by subtle changes in humidity, by the rise and fall of water levels in the vast wetlands that covered much of the landscape. The tidal cycles would have been more important than seasonal changes in many areas. Much of the Devonian landscape was influenced by tides, with vast areas of coastal wetlands that were alternately flooded and exposed as the tides changed. These tidal rhythms would have created a twice-daily cycle of inundation and exposure that would have been one of the most important environmental rhythms for many organisms. In this world, the great Prototaxites towers would have stood like silent sentinels, changing slowly with the passage of years and decades. Unlike modern trees, which show their age through annual growth rings, these fungal giants probably grew more continuously, adding layer upon layer of new tissue throughout the growing season. Their growth would have been visible over timescales that human observers would have found meaningful, with new height added noticeably from year to year, new branches developing at the crown, new reproductive structures appearing and disappearing with the cycles of spore production. The lifespan of individual towers is difficult to estimate, but some may have lived for centuries or even millennia. Like modern trees, they would have grown slowly but steadily, accumulating biomass year after year until they reached the physical limits imposed by their architecture and environment. The oldest towers would have been ancient monuments, standing since before the memory of any other organism in their ecosystem. The death of one of these giants would have been a significant event in the local ecosystem. Unlike modern trees, which fall with a crash and begin decomposing relatively quickly, a Prototaxites tower might have collapsed more gradually, its fungal tissue breaking down and returning its nutrients to the soil over a period of months or years. The death of a large tower would have created a temporary clearing in the landscape, a space where smaller organisms could establish themselves, where new towers might eventually rise to take the place of the old. But decomposition in the Devonian world would have been a different process than it is today. Without the complex communities of insects, bacteria, and other decomposers that break down dead plant material in modern ecosystems, the breakdown of organic matter would have been slower and less complete. This led to the accumulation of vast quantities of partially decomposed organic matter, creating the rich deposits of organic material that would eventually become coal seams. The slow decomposition also meant that nutrients were recycled more slowly through the ecosystem. Dead organic matter would accumulate in wetlands and shallow seas, creating thick layers of partially decomposed material that provided a rich source of nutrients for the fungi that specialized in breaking down organic matter. This abundant supply of decomposable material was one of the factors that allowed the fungal giants to grow so large. As the Devonian period progressed and moved toward its close, the first true trees began to appear on the landscape. These early trees, with names like Archaeopteris and Calixylon, started small but gradually grew taller and more complex. They represented a fundamentally different approach to the problem of being a large terrestrial organism, one based on photosynthesis rather than decomposition, on capturing sunlight rather than recycling organic matter. The appearance of the first trees marked the beginning of the end for the age of mushroom rulers. These early trees were still crude by modern standards, with simple branching patterns and basic vascular systems, but they had several crucial advantages over the fungal giants. They could grow faster once established, they could capture energy directly from sunlight, and most importantly, they began to cast shade. The rise of trees created a new kind of competition that fungi had never faced before. Trees could capture sunlight directly, converting it into energy through photosynthesis, with an efficiency that gave them a powerful advantage over organisms that had to rely on decomposing organic matter. As trees grew taller and their canopies spread wider, they began to dominate the landscape in a way that no organism had before, creating the first true forests in Earth's history. But the transition from fungal dominance to tree dominance wasn't sudden or dramatic. It took millions of years for trees to truly take over terrestrial ecosystems, and for much of that time, Prototaxites towers and early trees probably coexisted in the same landscapes, each occupying slightly different niches in the changing environment. The early trees were pioneers, colonizing areas where conditions were suitable for photosynthetic organisms. They preferred well-drained soils where their roots could penetrate deeply, and areas with good light exposure where their crowns could capture maximum sunlight. The fungal towers, meanwhile, continued to thrive in the wetland environments where they had always been most successful. But gradually, inevitably, the towering fungi found themselves in an increasingly shaded world. The thick canopies of early forest reduced the amount of sunlight reaching the ground, which affected the smaller plants that the fungi depended on for organic matter. The complex root systems of trees began to change soil chemistry and structure in ways that favoured their mycorrhizal fungal partners over the giant free-living fungi that had dominated the landscape for so long. The trees also began to change the physical environment in ways that were unfavourable to the fungal giants. Their deep root systems stabilised soils and reduced erosion, which reduced the supply of fresh organic matter that accumulated in wetland environments. Their transpiration created local climate effects that reduced humidity in some areas. Their fallen leaves changed the chemistry of decomposition, creating conditions that favoured different types of decomposer organisms. Climate change also played a role in the decline of the mushroom rulers. As the Devonian period ended and the Carboniferous began, Earth's climate started to shift in subtle but important ways. The warm, wet conditions that had favoured the giant fungi began to give way to more variable weather patterns. Ice ages became more common, though still brief by geological standards. Seasonal variations increased as the climate became less uniformly stable. The world was becoming more complex, more dynamic, more challenging for organisms that had evolved to thrive in the stable, predictable conditions of the mid-Devonian. The fungal giants, for all their impressive size and evolutionary success, were adapted to a world that was slowly disappearing. They had evolved during a time when being tall was enough, when the main challenge was simply reaching high enough to disperse spores effectively. But the new world emerging around them required different strategies, different approaches to the problems of survival and reproduction. The very success of the tree strategy began to undermine the environmental conditions that had allowed the fungal giants to thrive. Trees were more efficient at extracting nutrients from soil, leaving less available for decomposer organisms. They created more complex soil environments with distinct layers and microhabitats. They changed patterns of water flow and retention. In every way, they were reshaping the terrestrial environment to favour photosynthetic organisms over decomposer organisms. The last of the great Prototaxites towers probably stood sometime around 360 million years ago, as the Devonian period was drawing to a close and the Carboniferous was beginning. They didn't disappear all at once in some dramatic extinction event. Rather, they gradually became less common, less successful, smaller. New towers stopped appearing as frequently. Existing towers grew more slowly, reached less impressive heights, competed unsuccessfully with the rising trees for resources and space. The fossil record shows this gradual decline clearly. In early Devonian rocks, prototaxites fossils are common and large. By the Middle Devonian, they're still present but less abundant. By the Late Devonian, they're rare and small. By the early Carboniferous, they've disappeared entirely from the fossil record, marking the end of an era. Eventually, they faded from the fossil record entirely, leaving behind only their mysterious remains for us to puzzle over hundreds of millions of years later. Their disappearance marks the end of one of the most unusual chapters in the history of life on Earth, a time when fungi, not plants or animals, were the dominant large organisms on land. It was the end of an experiment that had lasted for tens of millions of years, an experiment in what it might mean to be a large terrestrial organism without being a plant. But the fungi didn't disappear. They simply adapted, evolved, found new niches in the changing world. Instead of competing with trees for height and visibility, they learned to work with them, forming the partnerships that we now know are essential to forest ecosystems. They became the invisible partners of the new forest world, the hidden networks that connected tree roots across vast distances, the recyclers that broke down fallen logs and dead leaves and returned their nutrients to the soil. They retreated from the visible world and became masters of the invisible one, building networks that were more complex and more resilient than any individual giant had ever been. In a sense, they traded individual monumentality for collective intelligence, trading the impressive sight of individual towers for the subtle power of vast underground networks that could span entire continents. This transition from giant individuals to distributed networks represents one of the most profound strategic shifts in the history of life. The fungal giants of the Devonian had achieved success through individual achievement, through growing larger and taller than any competitor. But their descendants learned a different lesson, that cooperation can be more powerful than competition, that resilience comes not from individual strength, but from collective intelligence, that the most lasting achievements might be the ones that support other forms of life rather than competing with them. Today, if you walk through any forest, you're walking through a fungal kingdom far more vast and complex than those Devonian towers ever were. Beneath your feet, extending through the soil in every direction for miles around, are networks of fungal threads called mycelia. These networks can extend for enormous distances, connecting trees across vast areas, sharing nutrients, water, and even information between different plants in ways that blur the boundaries between individual organisms. Scientists have discovered that these underground fungal networks function almost like a natural internet, allowing forests to communicate and cooperate in ways we're only beginning to understand. When a tree is attacked by insects, it can send chemical signals through the fungal network to warn neighbouring trees, which can then begin producing defensive compounds before the insects arrive. When an old tree is dying, it can transfer nutrients to younger trees through these same networks, essentially willing its resources to the next generation. The Wood Wide Web, as some researchers have started calling it, is in many ways more sophisticated than the individual giants of the Devonian. Instead of single massive structures reaching toward the sky, modern fungi have learned to build networks, communities, systems of cooperation that span entire ecosystems. They've learned that collaboration can be more powerful than competition, that resilience comes not from individual strength but from collective intelligence, and they're still experimenting, still innovating, still finding new ways to be fungi in an ever-changing world. In laboratories around the world, scientists are discovering new capabilities of fungal networks that challenge our understanding of intelligence and consciousness. Some fungi can solve maze puzzles, finding the shortest path between food sources with an efficiency that rivals the best computer algorithms. Others can build structures that respond to environmental changes, growing stronger when stressed or redirecting growth when they encounter obstacles. The slime mold Physarum polycephalum, for example, can solve complex rooting problems that challenge computer scientists. When food sources are placed at different locations in its environment, it can grow a network that connects all the food sources using the shortest possible total length of connections. This is a computationally difficult problem that requires sophisticated algorithms when solved by computers, but the slime mold solves it naturally through its growth patterns. Other fungi have been shown to have memory-like properties, changing their behaviour based on past experiences and environmental conditions. They can learn to avoid harmful chemicals, remember the locations of food sources, and even pass information about environmental conditions to their offspring through chemical signals. These abilities suggest forms of information processing and decision-making that we're only beginning to understand. There are even researchers working on what they call fungal technologies, using the natural growing patterns of fungi to create new materials for human use. Mycelium-based packaging that can replace plastic foam. Fungal leather that could replace animal products. Building materials grown from fungi that are stronger than many traditional options and completely biodegradable when their useful life is over. The company Ecovative has developed methods for growing packaging materials from mycelium that can replace polystyrene foam. The process involves feeding agricultural waste to fungi and allowing them to grow in specially designed molds. The resulting material is strong, lightweight, fire-resistant, and completely biodegradable. When the packaging has served its purpose, it can be composted in a backyard compost bin, returning its nutrients to the soil. Other companies are exploring the use of mycelium to create leather-like materials that could replace animal leather in shoes, bags, and clothing. These mycelium leathers can be grown in a matter of weeks, compared to the months or years required to raise animals for traditional leather. They can be engineered to have specific properties, made stronger or more flexible, or more water-resistant depending on the intended application. In a strange way, we might be witnessing the beginning of a new age of fungal innovation, a time when these ancient organisms are finding new roles to play in partnership with human technology. Not giants reaching toward the sky like their Devonian ancestors, but partners working with us to solve problems and build sustainable technologies. The descendants of those ancient towers might yet have important roles to play in shaping the future of our planet. But there's something deeper here. Something that goes beyond the specific story of prototaxites and early fungi. The world of the mushroom rulers reminds us that intelligence and innovation aren't limited to animals or even to individual organisms. The fungal networks beneath our feet are processing information, making decisions, solving problems in real time. They're just doing it in ways that we're only beginning to recognize and understand, in ways that challenge our assumptions about what it means to be intelligent, what it means to be conscious, what it means to be alive. This challenges our human-centered view of intelligence. We tend to think of intelligence as something that happens in brains, as something that requires neurons and synapses and electrical signals. But the fungal networks show us that intelligence can emerge from very different kinds of systems, from networks of simple connections that process information through chemical signals and growth patterns. The implications of this are profound. If intelligence can emerge from fungal networks, what other forms might it take? What other kinds of information processing and decision-making might be happening all around us, in systems that we don't recognize as intelligent because they don't look like brains or computers or human minds? When we walk through a forest today, we tend to focus on what we can see above ground. The trees reaching toward the sky, the flowers blooming in clearings, the birds moving through the branches, the sunlight filtering through leaves in constantly shifting patterns. But we're actually moving through a vast, mostly invisible civilization, a network of fungal minds that connects and coordinates an entire ecosystem. They're thinking in their own way. They're communicating across vast distances. They're making collective decisions about resource allocation, defense strategies, and growth patterns. They're solving problems that we're only beginning to understand, using methods that we're only starting to appreciate. They're demonstrating that intelligence doesn't require brains or consciousness as we usually think of it. The Devonian giants were impressive individuals, standing alone like monuments to their own success. They dominated the landscape through sheer size and persistence, reaching toward the sky with an ambition that wouldn't be matched until trees learned to do the same thing millions of years later. But their descendants became something more, something that might be more important in the long run. They became the living internet of the natural world, the connective tissue that holds ecosystems together, the infrastructure that makes complex ecological communities possible. They learned that cooperation can be more powerful than competition, that networks can be more resilient than towers, that sometimes the most important work happens invisibly, underground, in the spaces between other organisms. And perhaps there's a lesson in that transformation for us, as we build our own networks, our own technologies, our own ways of connecting across distances and sharing information. The fungi that once ruled the landscape by growing tall eventually found more elegant solutions. They learned to grow wide, to grow deep, to grow together. they discovered that the most lasting achievements might be the ones that support other forms of life, rather than competing with them. The spores that once drifted from those Devonian towers are still drifting today, carried by wind and water to new locations where they might take root and begin the ancient process of growth and connection all over again. In many ways, those ancient giants are still with us, not as fossils in museum displays, but as inspiration, as a reminder that life finds ways to innovate and adapt and persist, even when the world changes completely around it. Their legacy lives on in every forest, every garden, every handful of soil. Every time you see mushrooms sprouting after rain, every time you walk through a forest and breathe in the rich scent of decomposing leaves, every time you marvel at the way plants seem to communicate and cooperate, you're witnessing the continuation of experiments that began hundreds of millions of years ago in the age of mushroom rulers. As we near the end of our journey through this fungal world, it's worth remembering that you right now, wherever you're listening to this, are connected to this story in ways both visible and invisible. Your body is home to countless fungal partners, some helping with digestion, others protecting your skin from harmful bacteria, still others playing roles we don't fully understand yet. The human microbiome includes numerous fungal species that are essential to our health and well-being. The yeast that lives in our intestines helps break down food and produces vitamins that we can't make ourselves. The fungi on our skin form protective barriers against harmful bacteria and help maintain the chemical balance that keeps our skin healthy. Even our immune systems have learned to work with beneficial fungi while defending against harmful ones. The bread you might have eaten today was raised by yeast, fungal partners that have been working with humans for thousands of years. The wine or beer that people enjoy is the product of fungal fermentation, ancient biotechnology that transforms simple sugars into complex alcohols and flavors. The antibiotics that save lives are often produced by fungi, natural chemical weapons that fungi evolved to compete with bacteria and that we've learned to harness for our own purposes. The recycling of nutrients that keeps your local ecosystem functioning depends on fungal workers breaking down organic matter and making it available to new life. Every fallen tree, every dead animal, every pile of autumn leaves is processed by fungi that break down complex organic molecules and return their components to the soil where they can be used by living plants. You're living in a world shaped by fungi, a world where these organisms continue to innovate, adapt, and find new ways to connect life across time and space. The giants of the Devonian are gone, but their legacy lives on in every forest, every garden, every handful of soil. Their experiment in growing tall may have ended, but their experiment in connecting life continues, evolves, expands into new possibilities that we're only beginning to imagine. And maybe, just maybe, as you drift off to sleep tonight, you can imagine yourself walking through that ancient world. Picture yourself surrounded by pale towers reaching toward a warm, misty sky, breathing air thick with spores and possibility, standing at the edge of a time when Earth was young and life was still figuring out what it wanted to become. Feel the soft ground beneath your feet, spongy with moisture and teeming with invisible life. Listen to the gentle sounds of water dripping and wind moving through structures unlike anything in our modern world. Watch the play of light and shadow as the alien sun moves across a sky that knows no seasons, no harsh transitions, only the gentle rhythm of days that are slightly shorter than our own. The mushroom rulers are gone, but the kingdom they built, the network they pioneered, the partnerships they forged, these things persist and grow stronger with each passing year. And in their quiet way, they're still shaping our world, still connecting life in ways we're only beginning to understand, still building the invisible infrastructure that makes complex life on Earth possible. Sleep well, knowing that beneath the soil of this ancient planet, the descendants of those first fungal innovators are still at work, still growing, still connecting, still building bridges between life forms across the vast network of existence that connects every living thing on Earth. They're the quiet architects of our world, the invisible engineers who make the visible world possible. The more we learn about the Devonian world, the more we realize how profoundly different it was from anything we experience today. It wasn't just the absence of familiar organisms, the lack of birds or flowering plants or mammals. It was the entire rhythm of life, the fundamental patterns that governed how energy flowed through ecosystems, how information was processed and transmitted, how organisms related to their environment and to each other. In our modern world, we're used to thinking of ecosystems in terms of familiar categories: producers and consumers, predators and prey, competitors and mutualists. But the Devonian world operated according to different principles, different organizing forces. The massive fungal towers weren't just large organisms, they were ecosystem engineers, structures that created and maintained the environmental conditions that allowed other forms of life to flourish. The shade cast by a Prototaxites tower would have created microclimates beneath its canopy, areas where the humidity was higher, where the temperature was more stable, where smaller organisms could find refuge from the sometimes harsh conditions of the open landscape. These tower ecosystems would have been islands of biological diversity in what was otherwise a relatively simple landscape. The base of each tower would have been particularly rich in life. The constant rain of organic matter falling from the tower above, combined with the nutrients leaching from the fungal tissue itself, would have created soils that were extraordinarily fertile by Devonian standards. These nutrient-rich zones would have supported dense communities of smaller organisms, primitive plants, algae, bacteria, and the early arthropods that fed on them. We can imagine these tower bases as oases in the Devonian landscape, places where life was more abundant and more diverse than in the surrounding areas. They would have been the equivalent of modern forest clearings, except that instead of being created by fallen trees, they were actively maintained by living fungal giants that continued to shape their local environment for centuries or millennia. The ecological relationships within these tower ecosystems would have been unlike anything we see today. The fungi themselves would have been both habitat and organism, both environment and inhabitant. They created the conditions that supported other forms of life, but they also competed with those same organisms for nutrients and space. It was a delicate balance, maintained by evolutionary relationships that had no modern equivalent. Some of the smaller organisms living in these tower ecosystems might have been direct partners of the fungi, early examples of the kinds of symbiotic relationships that are so common in modern ecosystems. Primitive plants might have formed loose associations with fungal hyphae, receiving nutrients in exchange for organic compounds. Early arthropods might have helped disperse fungal spores in exchange for food or shelter, but many of these relationships would have been more opportunistic, more casual than the highly evolved partnerships we see in modern ecosystems. This was still early in the history of terrestrial life, when organisms were still learning how to live together, still working out the basic rules of ecological cooperation and competition. The process of discovering and understanding Prototaxites is itself a fascinating story that spans more than 150 years of scientific investigation. It's a tale that illustrates how science works, how our understanding of the past evolves as new tools and new perspectives become available, how even the most fundamental questions about life's history can remain mysteries for generations. The first Prototaxites fossil was discovered in 1859 by a Canadian geologist named John William Dawson. He found it in rocks along the shores of Gaspé Bay in Quebec, embedded in limestone that was clearly very old, but whose exact age wasn't yet understood. Dawson was struck by the size and strange structure of the fossil, but he had no context for interpreting what he was seeing. This was long before we understood plate tectonics, before we had developed radiometric dating techniques, before we even had a clear picture of the history of life on Earth. Charles Darwin had just published On the Origin of Species that same year, and the scientific community was still struggling to understand the basic principles of evolution and deep time. Dawson initially classified his find as a conifer, a primitive evergreen tree that he named Prototaxites logani. The name literally means first yew, reflecting his belief that he had discovered an early ancestor of modern coniferous trees. Given the limited knowledge available at the time, this was a reasonable interpretation. The fossil was clearly from some kind of large woody organism, and trees seemed like the most obvious explanation. But as more fossils were discovered over the following decades, it became clear that Prototaxites didn't fit neatly into any known category of plant. The internal structure was wrong for a tree. There was no evidence of the kinds of vascular tissues that plants use to transport water and nutrients. The growth patterns didn't match anything seen in modern or fossil plants. By the early 20th century, some scientists were beginning to suggest alternative interpretations. Perhaps these weren't trees at all, but some kind of giant algae that had adapted to terrestrial life. Or maybe they were an entirely extinct group of organisms, something that had no modern relatives and represented a failed experiment in the evolution of large terrestrial life. The mystery deepened as fossils were found on different continents, in rock formations of different ages. Prototaxites had clearly been a global phenomenon, lasting for tens of millions of years across multiple geological periods. Whatever these organisms were, they had been remarkably successful in their time. The debate continued for decades, with different scientists proposing different interpretations based on the limited evidence available. Some argued for a plant origin, others for algae, still others for entirely unknown groups of organisms. The fossils themselves were tantalizing but incomplete, showing the internal structure clearly but providing little information about the external appearance or the ecological context of these mysterious giants. It wasn't until the 1990s that new analytical techniques began to provide clearer answers. Advances in microscopy allowed scientists to examine the cellular structure of the fossils in unprecedented detail. Chemical analysis techniques made it possible to study the composition of the fossilized tissue, looking for clues about the biochemistry and metabolism of these ancient organisms. The breakthrough came when researchers began to compare the structure of Prototaxites fossils with modern organisms using these new analytical tools. The arrangement of cells, the composition of cell walls, the patterns of tissue organization, all pointed clearly toward a fungal origin. This wasn't just a larger version of a familiar organism, it was something entirely new, a form of life that had no modern equivalent. The realization that Prototaxites was a fungus created new mysteries, even as it solved old ones. How had fungi managed to grow so large? What ecological conditions had allowed them to become the dominant terrestrial organisms? Why had they disappeared, and what had replaced them? These questions opened up entirely new areas of research into the early history of terrestrial life. Modern research on Prototaxites continues to reveal new aspects of these remarkable organisms. Computer modelling of their internal structure suggests sophisticated systems for nutrient transport and structural support. Chemical analysis of their remains provides clues about their metabolism and their relationships with other organisms. Geological studies of the rock formations where they're found help reconstruct the environmental conditions in which they lived. One of the most intriguing recent discoveries is evidence that Prototaxites towers might not have been entirely fungal. Some researchers have found what appear to be the remains of other organisms embedded within the fungal tissue, suggesting that these giants might have been complex ecosystems in their own right, biological skyscrapers that provided habitat for entire communities of smaller organisms. This finding has led to new theories about the nature of these ancient giants. Perhaps they weren't just large fungi, but rather complex associations of different organisms that had learned to live together in ways that allowed them to achieve sizes that no single organism could reach alone. If true, this would make them even more remarkable. Early examples of the kinds of complex biological partnerships that are common in modern ecosystems. The study of Prototaxites has also provided new insights into the evolution of terrestrial ecosystems. By understanding how these early giants lived and died, how they interacted with their environment and with other organisms, scientists are beginning to piece together a picture of what life was like during the crucial early phases of terrestrial evolution. This research has implications that extend far beyond paleontology. Understanding how life first colonized the land, how the earliest terrestrial ecosystems were organized, how organisms adapted to the challenges of living outside of water, all of this provides context for understanding modern ecological processes and for predicting how ecosystems might respond to future environmental changes. The transition from the fungal giants to modern forest ecosystems represents one of the most profound ecological transformations in Earth's history. It wasn't just a change in which organisms were dominant, it was a fundamental shift in how terrestrial ecosystems were organized, how energy flowed through them, how nutrients were cycled, how information was processed and transmitted. The world of the mushroom rulers was organized around decomposition and recycling. The great fungal towers were essentially biological recycling plants, breaking down organic matter and returning nutrients to the system. Energy flowed primarily through decomposer organisms, with photosynthesis playing a supporting role. This created ecosystems that were remarkably stable and resilient, but also relatively simple in their organization. The rise of trees created a fundamentally different kind of ecosystem, one organized around photosynthesis and primary production. energy began to flow primarily through green plants, with decomposer organisms playing a supporting role. This allowed for much more complex food webs, more diverse communities of organisms, and more dynamic ecological relationships. But the transition wasn't just about which organisms were dominant. It also represented a shift from ecosystems based on cooperation to ecosystems based on competition. The fungal giants had succeeded through their ability to work together, to form networks, to share resources and information. Trees succeeded through their ability to compete for light, water, and nutrients, to grow faster and taller than their neighbours. This shift from cooperation to competition, as the primary organising principle of terrestrial ecosystems, had profound implications for the evolution of life on Earth. It created the selective pressures that drove the rapid diversification of plant and animal life that characterised the later Paleozoic era. It set the stage for the evolution of complex behaviours, sophisticated sensory systems, and eventually intelligence and consciousness. But the cooperative strategies pioneered by the fungal giants didn't disappear. They went underground, literally, becoming the foundation of the mycorrhizal networks that connect modern forests. In a sense, the age of mushroom rulers never really ended. It just became invisible, operating beneath the surface of ecosystems that appear to be dominated by competitive interactions. This hidden cooperative infrastructure is what makes modern forests possible. The trees that appear to be competing with each other for light and space are actually connected by vast underground networks that allow them to share resources, communicate about threats, and support each other through difficult periods. The competitive surface layer of the ecosystem is supported by a cooperative underground layer that traces its origins back to the age of mushroom rulers. Understanding this dual nature of modern ecosystems, the interplay between competitive and cooperative processes has become one of the most important frontiers in ecological research. It's changing how we think about conservation, about restoration, about the management of natural resources. It's revealing that ecosystems are far more sophisticated and interconnected than we previously understood. The more we learn about fungal networks, the more we realize that they represent a form of natural intelligence that operates according to principles very different from the kinds of intelligence we're familiar with. They process information, make decisions, solve problems, but they do it through chemical signals and growth patterns rather than through electrical impulses and neural networks. This has led some researchers to suggest that fungal networks might represent a form of distributed intelligence, a way of processing information and making decisions that doesn't require a central control system. Instead of having a brain that coordinates all activities, the network itself becomes the intelligence, with decision-making emerging from the interactions between different parts of the system. This kind of distributed intelligence might be more common in nature than we previously thought. Social insect colonies, flocks of birds, schools of fish, all of these represent systems where intelligent behaviour emerges from the interactions between simple individual agents. Fungal networks might be another example of this phenomenon, operating on a much larger scale and over much longer time periods than the examples we're more familiar with. The implications of this for our understanding of intelligence and consciousness are profound. If intelligence can emerge from fungal networks, from social insect colonies, from other forms of distributed systems, then perhaps consciousness isn't as rare or as special as we like to think. Perhaps it's a more general property of complex systems, something that can emerge whenever information is processed and decisions are made, regardless of the specific physical substrate. This doesn't diminish the importance or uniqueness of human consciousness, but it does place it in a broader context. We're not the only intelligent beings on this planet. We're part of a larger community of minds that includes many forms of life that we're only beginning to understand and appreciate. The fungal networks beneath our feet are thinking in their own way. They're processing information about soil conditions, water availability, nutrient distribution, and the health and needs of their plant partners. They're making decisions about where to grow, how to allocate resources, when to reproduce. They're solving problems that affect the health and stability of entire ecosystems. And they're doing all of this without consciousness as we understand it, without self-awareness, without the kinds of mental experiences that characterize human cognition. This suggests that intelligence might be much more widespread in nature than we thought, that it might be a fundamental property of complex living systems, rather than a rare accident that occurred only in a few species with large brains. The study of fungal intelligence is still in its early stages, but it's already beginning to influence how we think about artificial intelligence and machine learning. Computer scientists are studying fungal networks to understand how distributed systems can process information and make decisions without central control. Engineers are looking at fungal growth patterns for inspiration in designing self-organizing materials and structures. The biomimetic potential of fungal systems is enormous. Their ability to grow efficient networks, to adapt to changing conditions, to process information through chemical signals, all of these represent technologies that could have applications in everything from computer science to architecture to materials engineering. But perhaps the most important lesson we can learn from the fungi is about the power of cooperation and partnership. The great fungal towers of the Devonian succeeded not through individual achievement, but through their ability to work together, to form networks, to support each other. When conditions changed and individual giants were no longer viable, they adapted by becoming even more cooperative, forming the partnerships with plants that are still essential to terrestrial ecosystems today. This suggests that cooperation, not competition, might be the more fundamental principle underlying the organization of life. Competition gets more attention because it's more visible, more dramatic, more easily studied. But cooperation often operates in the background, providing the foundation that makes competition possible. In human society, we often focus on competitive processes, on individual achievement, on the importance of standing out from the crowd. But perhaps we could learn something from the fungi about the power of networks, about the importance of cooperation, about the value of supporting each other rather than just competing with each other. The age of mushroom rulers ended 350 million years ago, but their legacy continues in the underground networks that support modern ecosystems. They remind us that cooperation can be just as powerful as competition, that intelligence can take many different forms, that the most important work is often invisible, happening behind the scenes where few people notice it. As we face the challenges of the modern world, climate change, environmental degradation, social inequality, perhaps we could benefit from thinking more like fungi, building networks instead of walls, sharing resources instead of hoarding them, supporting each other instead of trying to outcompete each other. The mushroom rulers understood something about survival that we're still learning, that in the long run, we're all connected, and our success depends on the health of the whole system, not just our individual achievements. The Devonian world might seem alien to us, with its towering fungi and primitive plants, its warm, humid climate and strange atmospheric composition. But in many ways, it was a more cooperative world than our own, a world where the largest organisms succeeded through partnership rather than competition, where information was shared freely through vast networks, where individual success was inseparable from the health of the community. There's something beautiful about imagining that world, something peaceful about thinking of those great towers standing in their ancient landscapes, connected to each other through invisible networks, sharing resources and information across vast distances, supporting the small communities of organisms that lived in their shadows. It was a world that worked differently than ours, but it worked remarkably well for tens of millions of years. And perhaps, as you settle in for sleep tonight, you can carry some of that peacefulness with you. Imagine yourself in that ancient world, surrounded by the gentle giants of the Devonian, feeling the warm, humid air on your skin, hearing the soft sounds of that primordial forest, knowing that you're part of a vast network of life that connects every living thing. The fungal networks beneath your feet right now are descendants of those ancient systems, carrying forward the same patterns of cooperation and support that allowed life to flourish in that distant world. You're connected to that ancient story, part of a continuous thread that stretches back through hundreds of millions of years to the age of mushroom rulers. Sleep well, knowing that the principles that guided those ancient giants, cooperation, partnership, mutual support, continue to operate in the world around you. The age of mushroom rulers may be over, but the age of cooperation continues, and you're part of it. The networks that connect us all, visible and invisible, ancient and modern, are still growing, still supporting life, still creating the conditions that make everything else possible. There's something profound about the way time works in the fungal world that challenges our human understanding of duration and permanence. We live in a world of quick changes, of daily rhythms and seasonal cycles, of lifespans measured in decades rather than centuries. But the fungal networks that descended from those ancient giants operate on entirely different temporal scales, growing and changing over periods that span human generations, making decisions and adjustments that unfold over decades or centuries. A mycorrhizal network in a modern forest might be hundreds or even thousands of years old, older than any individual tree it connects, older than most human civilizations. These networks have witnessed the rise and fall of countless individual organisms, the slow succession of forest communities, the gradual changes in climate and environment that occur over geological timescales. They represent a form of institutional memory, a living record of environmental conditions and ecological relationships that stretches back through centuries of forest history. This long-term perspective gives fungal networks a kind of wisdom that emerges from their ability to integrate information over vast periods of time. They respond not just to immediate conditions, but to long-term trends, to patterns that emerge over decades or centuries. When a forest is stressed by drought or disease, the fungal networks can draw on centuries of experience to guide their responses, allocating resources based on patterns they've learned over their extended lifespans. This temporal perspective is something we humans struggle to understand or emulate. Our decision-making is dominated by short-term concerns, by immediate pressures and urgent needs. We have difficulty thinking beyond the next few years or decades, let alone the centuries or millennia that represent meaningful timescales from an ecological perspective. Perhaps this is one of the most important lessons we can learn from the fungi, the value of long-term thinking, of making decisions based on patterns that emerge over extended periods rather than just immediate circumstances. The chemistry of the Devonian atmosphere would have created conditions unlike anything in our modern experience. The high carbon dioxide levels wouldn't just have affected photosynthesis and temperature, they would have changed the fundamental chemistry of life itself. Carbonic acid, formed when carbon dioxide dissolves in water, would have made the rains slightly acidic, accelerating the weathering of rocks and increasing the availability of mineral nutrients in the soil. This enhanced weathering would have been crucial for the early terrestrial ecosystems, providing the mineral nutrients that plants and fungi needed to grow. The combination of high carbon dioxide levels and enhanced weathering created a kind of natural fertilization system that supported the rapid growth and large size of organisms like prototaxites. It was a world where the atmosphere itself was more nutritious, where the basic building blocks of life were more readily available than they are today. The isotopic composition of the Devonian atmosphere also tells us something about the global cycles that were operating at the time. Carbon isotope ratios preserved in ancient rocks suggest that the carbon cycle was operating very differently than it does today, with much larger reservoirs of organic carbon being stored in soils and sediments. This massive carbon storage was partly responsible for the eventual decline in atmospheric carbon dioxide levels that marked the end of the greenhouse conditions of the Devonian. The role of fungi in this global carbon cycle would have been enormous. As the primary decomposers in terrestrial ecosystems, they were responsible for breaking down organic matter and releasing carbon dioxide back to the atmosphere. But they were also responsible for creating and maintaining the organic-rich soils that stored vast quantities of carbon. The balance between these two processes, decomposition and storage, would have been a crucial factor in determining the composition of the atmosphere and the stability of the climate. Understanding this ancient carbon cycle has important implications for our modern world. As we grapple with rising carbon dioxide levels and climate change, we can learn from the natural processes that regulated Earth's climate in the past. Fungi were key players in these ancient regulatory systems, and they may be important allies in our efforts to stabilize the modern climate. Modern research into fungal carbon storage is revealing that these organisms play a much larger role in the global carbon cycle than previously thought. Mycorrhizal fungi can store significant amounts of carbon in their tissues and in the soil organic matter they help create. Some estimates suggest that fungal networks store as much carbon as all the plants in terrestrial ecosystems combined. This hidden carbon reservoir represents a crucial component of Earth's climate system that we're only beginning to understand. The preservation of prototaxites fossils tells its own remarkable story about the conditions that existed in the Devonian world. Most organic material decomposes completely within years or decades of an organism's death, leaving no trace in the geological record. The The fact that we can find detailed fossils of these fungal giants hundreds of millions of years later tells us something important about the environments in which they lived and died. The best preserved Prototaxites fossils are found in limestone formations that were deposited in shallow marine environments. This suggests that many of these giants lived in coastal areas, perhaps in tidal zones or near river mouths where terrestrial and marine environments met. When they died, they were quickly buried by marine sediments that protected them from decay and preserved their internal structure in remarkable detail. The process of fossilization itself would have been complex and unusual for these organisms. Unlike the bones of animals or the woody tissue of trees, fungal tissue is soft and cellular, not naturally inclined toward preservation. The fact that it fossilized at all suggests very specific chemical conditions in the sediments where these organisms were buried, conditions that allowed the replacement of organic tissue with minerals while preserving the original structure. Some of the most spectacular Prototaxites fossils show not just the internal structure of the organisms, but also evidence of their interactions with other forms of life. There are fossils that show boreholes made by early arthropods, suggesting that these giants served as habitat for smaller organisms even while they were alive. Other fossils show evidence of bacterial or algal growth on the surfaces of the towers, creating complex biological communities centered around the fungal giants. These ecological interactions preserved in the fossil record give us glimpses of the complex relationships that existed in Devonian ecosystems. They suggest that the fungal towers weren't just isolated giants, but were integrated into networks of biological relationships that included many different types of organisms. They were ecosystem engineers in the truest sense, creating and maintaining environmental conditions that supported entire communities of life. The distribution of Prototaxites fossils around the world also tells us about the paleogeography of the Devonian period. These organisms are found on every continent, but their abundance and size vary significantly from place to place. This suggests that while they were globally distributed, they thrived best in certain types of environments and struggled in others. The largest and most abundant fossils are found in what were tropical or subtropical regions during the Devonian, areas that experienced warm, humid conditions year-round. Smaller, less common fossils are found in what were higher latitudes, suggesting that these organisms were most successful in the warm, stable climates that characterize the equatorial regions of the Devonian world. This geographic distribution provides important clues about the environmental requirements of these ancient giants. They clearly needed warm, humid conditions to reach their full potential, which makes sense given their fungal nature. Modern fungi are generally most successful in warm, moist environments where organic matter is abundant and decomposition proceeds rapidly. The study of Prototaxites has also contributed to our understanding of how fossilization works and how the geological record preserves evidence of ancient life. These fossils represent one of the best examples we have of soft tissue preservation from the early Paleozoic era. By studying how fungal tissue was preserved under ancient conditions, paleontologists have developed better techniques for finding and interpreting other types of soft tissue fossils. This has led to discoveries of other unusual organisms from the Devonian period, including primitive animals and plants that were previously unknown to science. The techniques developed for studying Prototaxites have opened new windows into the early history of terrestrial life, revealing a world that was far more diverse and complex than we previously understood. The implications of this research extend beyond paleontology into fields like astrobiology and the search for life on other planets. Understanding how life colonized land on Earth, how early terrestrial ecosystems were organized, how organisms adapted to the challenges of living outside of water, all of this provides context for thinking about how life might evolve on other worlds with different environmental conditions. The fungal giants of the Devonian represent a solution to the challenges of terrestrial life that was very different from the plant-dominated ecosystems we're familiar with today. This suggests that there might be multiple viable approaches to organizing terrestrial ecosystems, multiple ways that life could adapt to the challenges of living on the land surface of a planet. If we ever discover life on other worlds, it might look more like the Devonian fungal forests than like our modern plant-dominated landscapes. Understanding how these ancient ecosystems worked, how they were organized, how they processed energy and information, might give us better tools for recognizing and understanding alien forms of life. The transition from the age of mushroom rulers to modern forest ecosystems also provides insights into how ecosystems respond to environmental change. The Devonian period ended with a series of extinction events that dramatically altered terrestrial life, creating new environmental conditions that favoured different types of organisms. The fungal giants were among the casualties of these changes, but their decline wasn't sudden or catastrophic. Instead, it was a gradual process that took place over millions of years, as environmental conditions slowly shifted and new competitors arose. This provides a model for understanding how modern ecosystems might respond to ongoing environmental changes like climate change and habitat destruction. The resilience of the fungal networks themselves, their ability to adapt and persist even as the surface communities they supported changed dramatically, suggests that some aspects of ecosystems are more stable and persistent than others. The underground networks that connected the Devonian giants survived the transition to tree-dominated forests and continue to play crucial roles in modern ecosystems. This has important implications for conservation and ecosystem management. It suggests that protecting the invisible infrastructure of ecosystems, the fungal networks and microbial communities that operate below ground, might be just as important as protecting the more visible plants and animals that attract most of our attention. Modern conservation efforts are beginning to recognise the importance of soil communities and underground networks. There are now programs focused specifically on protecting mycorrhizal fungi and promoting the health of soil ecosystems. These programs recognise that the above-ground communities we see are supported by below-ground communities that are often far more diverse and complex. The study of ancient fungal ecosystems like those of the Devonian period provides crucial context for these modern conservation efforts. By understanding how these systems have changed over geological timescales, how they've responded to past environmental changes, how they've adapted and evolved, we can better predict how they might respond to future changes and better design strategies to protect them. The biochemistry of the ancient fungal giants would have been fascinating and complex, involving metabolic pathways that we can only partially reconstruct from the fossil evidence. These organisms were processing enormous quantities of organic matter, breaking down complex molecules and reassembling them into new forms, cycling nutrients and energy through their towering structures at a scale that dwarfed anything else in their environment. The enzymatic systems required for this large-scale decomposition would have been incredibly sophisticated, involving hundreds or thousands of different enzymes working in coordinated networks to break down everything from simple sugars to complex lignin-like compounds. These biochemical networks would have represented some of the most complex chemical processing systems on the planet at the time, biological factories that could rival modern industrial processes in their efficiency and scope. Modern studies of fungal biochemistry reveal the extraordinary diversity and sophistication of the chemical processes that these organisms can carry out. Fungi produce thousands of different enzymes, many of which have applications in biotechnology and industrial processes. They can break down virtually any organic compound, from simple carbohydrates to complex pollutants, making them valuable allies in everything from waste treatment to environmental remediation. The ancient giants would have possessed even more impressive biochemical capabilities, given their size and the diversity of organic matter they would have encountered in their environment. They would have needed to process plant matter, animal remains, bacterial and algal biomass, and probably many types of organic compounds that no longer exist in modern environments. This biochemical versatility would have made them incredibly efficient recyclers, able to extract nutrients from virtually any organic source and convert it into new fungal tissue or return it to the soil for use by other organisms. They were the ultimate generalists, able to thrive in any environment where organic matter was available for decomposition. The loss of these biochemical capabilities when the giants disappeared would have had profound effects on ecosystem functioning. The early trees that replaced them as the dominant terrestrial organisms had much more limited abilities to process organic matter. This led to the accumulation of partially decomposed plant material that eventually became coal deposits, representing a fundamental change in how carbon was cycled through terrestrial ecosystems. Understanding these ancient biochemical systems has practical applications for modern biotechnology. Many of the enzymes that the fungal giants would have used are still present in their modern descendants, though often in less active or diverse forms. By studying these enzymes and understanding how they evolved, scientists can develop new tools for industrial applications, environmental remediation, and biotechnology. The sensory world of the Devonian giants would have been unlike anything we can easily imagine. These organisms would have experienced their environment through chemical gradients and molecular signals rather than through sight, sound, or touch as we understand them. They would have been constantly sampling their chemical environment, detecting the presence of nutrients, competitors, partners, and threats through sophisticated chemical sensing systems. This chemical communication would have operated over multiple scales, from local interactions within individual towers to long-distance signals transmitted through soil networks across entire landscapes. The fungal giants would have been both sending and receiving chemical messages constantly, creating a landscape-scale communication network that coordinated growth, reproduction, and resource allocation across vast areas. The complexity of this chemical communication system would have been staggering. Different types of chemical signals would have carried different types of information, using molecular languages that we're only beginning to understand in modern fungi. There would have been signals for identifying species and individuals, signals for coordinating reproductive activities, signals for warning about threats or competing for resources. This chemical landscape would have been as rich and complex as the visual and auditory landscapes that we're more familiar with, but operating according to principles that are largely foreign to our human senses. The giants would have experienced a world of molecular information, chemical gradients, and biochemical signals that would have been invisible to organisms that relied primarily on vision and hearing. Modern research into fungal communication is revealing the sophistication of these chemical signaling systems. Fungi can produce and respond to hundreds of different chemical compounds, using them to coordinate complex behaviors across entire networks. They can send specific chemical messages to particular species or individuals, broadcast general warnings across wide areas, or create chemical trails that guide growth and development. The implications of this for our understanding of consciousness and intelligence are profound. If fungi can process complex information, make sophisticated decisions, and coordinate complex behaviors using chemical signaling systems, this suggests that intelligence might be much more widespread in nature than we previously thought. It challenges our assumptions about what kinds of physical systems are capable of intelligent behavior. The emotional landscape of learning about these ancient giants is complex and layered. There's a sense of wonder at discovering these remarkable organisms that dominated the Earth for tens of millions of years, a feeling of awe at their size and sophistication, and perhaps a touch of sadness at their loss. But there's also something deeply comforting about understanding our connection to these ancient systems, about recognizing that we're part of a continuous story that stretches back through deep time. The realization that the descendants of these giants are still with us, still supporting the forests we walk through, still processing the nutrients that feed the plants we depend on, creates a sense of continuity and connection that transcends the apparent discontinuity of geological time. We're not separate from these ancient stories, we're part of them, connected through invisible networks that link us to forms of life that existed hundreds of millions of years before humans appeared on the scene. This connection challenges the human tendency to see ourselves as separate from nature, as somehow standing outside the natural systems that surround us. The fungal networks beneath our feet remind us that we're embedded in living systems that are far older and far more complex than human civilization, systems that have been operating according to their own logic for hundreds of millions of years. There's something humbling about this realization, but also something liberating. It suggests that we don't have to solve all the world's problems by ourselves, that we're part of larger systems that have their own intelligence, their own capacity for adaptation and problem solving. The networks that supported the Devonian giants are still operating, still experimenting, still finding new ways to connect life across time and space. As our understanding of these ancient ecosystems deepens, we're beginning to realize that the transition from the age of mushroom rulers to modern forest ecosystems might not be as complete as we once thought. In some environments, fungi still play dominant roles, creating ecosystems that might be closer to the Devonian world than to typical modern forests. In some boreal forests, for example, the biomass of fungi exceeds that of all other organisms combined. These fungal-dominated ecosystems process nutrients and energy in ways that are reminiscent of the ancient giants, though on a smaller scale and in partnership with modern plants rather than as independent towers. There are also environments where fungi create structures that echo the architectural achievements of their ancient relatives. Some mycorrhizal networks create massive underground structures that can span hundreds of acres, connecting thousands of individual plants in networks that process information and resources on a scale that rivals the ancient towers. These modern fungal achievements suggest that the potential for fungal dominance hasn't disappeared, it's just been channeled into different forms. The descendants of the Devonian giants haven't given up their ambitions for large-scale ecosystem engineering, they've just found new ways to express those ambitions that are compatible with the tree-dominated world they now inhabit. This gives us reason to hope that some of the ecological principles that made the Devonian world so stable and resilient might still be operating in modern ecosystems. The cooperative networks, the long-term thinking, the sophisticated information processing capabilities that characterize the age of mushroom rulers are still present, still working to maintain the health and stability of the natural world. Understanding these continuities between ancient and modern ecosystems is becoming increasingly important as we face the challenges of environmental change and ecosystem management. The principles that allowed life to thrive during the age of mushroom rulers, cooperation over competition, long-term stability over short-term gain, network effects over individual achievement, might be exactly what we need to create more sustainable and resilient human societies. The study of these ancient giants also provides perspective on the current biodiversity crisis and the ongoing sixth extinction. The Devonian world was home to forms of life that seem almost impossibly exotic by modern standards, organisms that had found ways of being alive that have no modern equivalent. The loss of these ancient life forms represents a permanent reduction in the diversity of solutions that life has found to the challenges of existence. But the fact that some aspects of these ancient systems have persisted, that the cooperative networks and information processing capabilities of the fungal giants have found new expression in modern ecosystems, suggests that not all innovations are lost when species go extinct. Some capabilities, some ways of organizing life, can persist even when the specific organisms that first developed them disappear. This has important implications for how we think about conservation and extinction. It suggests that protecting ecosystem functions might be just as important as protecting individual species, that maintaining the networks and relationships that connect different forms of life might be crucial for preserving the capabilities that those networks represent. The underground world that exists beneath our feet right now is in many ways a continuation of the networks that supported the Devonian giants. These modern networks are processing information, making decisions, solving problems, supporting the visible ecosystems that we're more familiar with. They represent a form of natural intelligence that has been operating continuously for hundreds of millions of years, adapting and evolving but never disappearing. As you prepare for sleep tonight, you can take comfort in knowing that you're surrounded by these ancient networks, that the principles of cooperation and mutual support that characterize the age of mushroom rulers are still operating in the world around you. The fungal networks beneath the soil are still working, still growing, still connecting life across vast distances, still building the invisible infrastructure that makes the visible world possible. There's something deeply peaceful about understanding your place in these ancient stories, about recognizing that you're part of a continuous thread of life that stretches back through deep time to the age of mushroom rulers and forward into an unknown future. The networks that connect us all, the partnerships that support us, the quiet cooperation that makes everything else possible, these things are older than mountains, older than continents, older than most of the life forms we're familiar with. Sleep well, knowing that the wisdom of the ancient giants, their understanding of cooperation and long-term thinking, their sophisticated approaches to information processing and resource sharing continue to operate in the world around you. The age of mushroom rulers may be over, but their legacy lives on in every forest, every garden, every handful of soil, still growing, still connecting, still supporting the magnificent diversity of life that surrounds us. The diversity within the genus Prototaxites itself tells a fascinating story of evolutionary experimentation and adaptation. While we often speak of these organisms as if they were all identical, the fossil record reveals a remarkable variety of forms, sizes and structural arrangements that suggest millions of years of evolutionary refinement and adaptation to different environmental conditions. Some species of prototaxites were relatively modest in size, growing only to heights of 6 or 8 feet, more like large shrubs than the towering giants we usually picture. These smaller forms might have occupied different ecological niches, perhaps specializing in different types of substrates or thriving in environments where the full-size giants couldn't establish themselves successfully. Other species reached truly enormous proportions, with some fossil evidence suggesting individuals that might have grown to heights of 30 feet or more, dwarfing even the larger specimens we typically discuss. These mega-giants would have been among the largest living structures ever to exist on land, biological monuments that would have dominated their landscapes in ways that are difficult for us to comprehend. The structural variations between species were equally remarkable. Some developed elaborate branching patterns, creating candelabra-like forms with multiple growing tips that would have maximized their spore dispersal capabilities. Others remained largely unbranched, growing as single, massive columns that reached toward the sky with singular determination. Still others developed bulbous, almost club-like forms that suggest different strategies for resource storage and reproductive timing. The internal architecture varied significantly between species as well. Some developed very fine, dense networks of internal channels, suggesting highly efficient nutrient transport systems. Others had larger, more widely spaced chambers that might have served different functions, perhaps providing structural support for the enormous weight of the organism or creating specialized environments for different types of metabolic processes. These variations suggest that the prototaxite's lineage was actively experimenting with different solutions to the fundamental challenges of being a large terrestrial organism. Each species represented a different approach to the problems of structural support, nutrient transport, reproduction, and environmental adaptation. It was evolution in action, testing multiple strategies simultaneously across millions of years of geological time. The geographic distribution of different species also provides clues about the environmental conditions that favored different forms. The largest species tend to be found in fossil deposits that represent tropical or subtropical environments, areas where warm, humid conditions and abundant organic matter would have supported maximum growth. Smaller, more branched forms are often found in deposits that suggest more marginal environments, perhaps areas where resources were less abundant or environmental conditions were more challenging. This pattern suggests that the fungal giants were sensitive to environmental gradients in ways that allowed them to adapt their growth strategies to local conditions. Like modern fungi, they were probably able to modify their development in response to nutrient availability, moisture levels, and competition from other organisms. This phenotypic plasticity would have been crucial for their success across the diverse environments of the Devonian world. The study of these species-level differences has been hampered by the challenges of working with such ancient fossils. Many specimens are incomplete, and the processes of fossilization can obscure the subtle differences that distinguish one species from another. But as analytical techniques improve and more fossils are discovered, we're beginning to develop a clearer picture of the remarkable diversity that existed within this group of organisms. Recent advances in digital reconstruction techniques have allowed paleontologists to create three-dimensional models of prototaxites fossils, revealing structural details that were previously impossible to study. These digital reconstructions show that the internal architecture of these organisms was even more complex than previously thought, with sophisticated systems of channels, chambers, and support structures that rival the engineering of modern skyscrapers. The implications of this structural sophistication are profound. It suggests that these organisms had evolved highly refined solutions to the engineering challenges of building large biological structures, solutions that in some cases might be superior to anything we've achieved with modern materials and construction techniques. The study of these ancient biological architectures is beginning to influence modern engineering and materials science, providing inspiration for new approaches to building large, efficient, and sustainable structures. The seasonal cycles in the Devonian world, while gentler than those we experience today, would still have created rhythms that influenced the growth and reproduction of the fungal giants. Even in the warm, stable climate of the time, there would have been subtle variations in temperature, humidity, and daylight that mark the passage of the years and provided cues for biological processes. The fungal towers would have responded to these seasonal signals in ways that we can only partially reconstruct from the fossil evidence. Growth might have been fastest during certain periods when conditions were optimal, perhaps during times when rainfall was highest, or when the decomposition of organic matter was most active. Reproduction might have been timed to coincide with favourable conditions for spore dispersal, perhaps during periods when wind patterns were most consistent, or when atmospheric conditions provided the best opportunities for long-distance transport. The timing of these biological cycles would have been crucial for the success of individual organisms and for the coordination of reproductive activities across entire populations. Synchronized spore release across large numbers of towers would have maximized the chances of successful fertilization and colonization of new areas. This kind of population-level coordination suggests sophisticated communication systems that could operate across vast distances and timescales. Modern fungi show remarkable abilities to coordinate their activities across large networks, timing reproductive events to coincide with optimal conditions and synchronizing their responses to environmental changes. The Devonian giants would have needed even more sophisticated coordination systems, given their size and the timescales over which they operated. A single tower might have taken decades to mature, requiring long-term coordination with environmental cycles and with other members of its population. The death and decomposition of these giants would have been spectacular events that shaped their local environments for years or decades after the organism died. Unlike modern trees, which decompose relatively quickly once they fall, the massive fungal towers would have broken down slowly, their tough fungal tissue resisting decay and gradually releasing nutrients back to the soil over extended periods. This slow decomposition would have created unique microhabitats and nutrient gradients that supported specialized communities of organisms. The slowly decaying towers would have been like biological time-release capsules, gradually making their accumulated resources available to other forms of life over months or years. These decomposition sites might have been oases of biological activity, supporting higher densities and greater diversity of life than the surrounding areas. The chemical compounds released during the decomposition of fungal tissue would have been complex and varied, including many substances that don't exist in significant quantities in modern ecosystems. Some of these compounds might have served as nutrients for other organisms, while others might have acted as signaling molecules or defensive chemicals. The decomposition of a large Prototaxites tower would have been a major chemical event, altering the local environment in ways that could persist for decades. Understanding these decomposition processes has implications for modern ecology and biogeochemistry. Fungi play crucial roles in nutrient cycling in modern ecosystems, but the scale and intensity of these processes during the age of mushroom rulers would have been unlike anything we see today. Studying how these ancient systems worked might provide insights into more efficient and sustainable approaches to waste processing and nutrient management. The arthropod communities that lived alongside the fungal giants were remarkable in their own right, representing some of the earliest experiments in terrestrial animal life. These creatures were pioneers, solving fundamental problems of land-based existence that their descendants would build upon for hundreds of millions of years. But they were also intimately connected to the fungal towers, depending on them for food, shelter, and the environmental conditions that made terrestrial life possible. Some of the early arthropods were direct associates of the towers, living on or within the fungal tissue and feeding on the organic matter that the towers provided. Fossil evidence shows boreholes and tunnels within Prototaxites fossils, suggesting that some arthropods had developed the ability to excavate burrows within the living towers, creating sheltered environments where they could complete their life cycles. These boring arthropods would have been ecosystem engineers in their own right, creating microhabitats within the towers that might have been colonized by other organisms. The burrows they created could have served as nurseries for smaller arthropods, as refuges during adverse weather conditions, or as sites for specialized forms of decomposition that broke down fungal tissue in new ways. Other arthropods were more casual associates of the towers, using them as landmarks for navigation, as elevated platforms for surveillance or mating displays, or as sources of chemical cues that helped them locate food or avoid predators. The towers would have been prominent features in the relatively flat landscapes of the Devonian world, visible from great distances and serving as focal points for animal activities. The chemical ecology of these relationships would have been extraordinarily complex. Fungi are master chemists, producing hundreds or thousands of different compounds for communication, defense, and metabolic regulation. The arthropods living in association with the towers would have been exposed to this rich chemical environment, potentially developing specialized adaptations for detoxifying harmful compounds or utilizing beneficial ones. Some arthropods might have evolved symbiotic relationships with the fungi, receiving protection or nutrients in exchange for services like spore dispersal or defense against competing organisms. These early experiments in inter-kingdom cooperation would have been prototypes for the more sophisticated symbioses that characterize modern ecosystems. The predatory arthropods of the Devonian world were particularly fascinating, representing some of the first land-based predators in Earth's history. These creatures had to solve fundamental problems that had never been encountered before: how to hunt in three-dimensional space, how to locate prey using chemical and mechanical senses rather than the lateral line systems that worked in water, how to capture and subdue prey without the resistance of water to help with the mechanics of feeding. Some of these early predators were quite large by modern standards, with fossil evidence of arachnids that could exceed a foot in length, and millipede-like creatures that might have grown to several feet long. These giants would have been the apex predators of their terrestrial ecosystems, playing ecological roles similar to those filled by large vertebrates in modern ecosystems. The hunting strategies employed by these early predators would have been constrained by the limited sensory and locomotory capabilities available to arthropods. Without the sophisticated visual systems of vertebrates, or the complex behavioral repertoires that characterize modern predators, they would have relied primarily on chemical detection and mechanical vibration to locate prey. This would have created unique predator-prey dynamics that have no modern analogues. The impact of these predatory arthropods on the fungal towers and their associated communities would have been significant. Predation pressure would have shaped the evolution of defensive strategies in smaller arthropods, potentially driving the development of the boring behaviors that allowed some species to find refuge within the towers. It might also have influenced the chemical defenses of the fungi themselves, favoring towers that could produce compounds that deterred arthropod herbivores. The evolutionary arms race between predators and prey in these early terrestrial ecosystems would have been a driving force for innovation and diversification. Each new predatory strategy would have created selective pressure for new defensive adaptations, leading to escalating cycles of evolutionary innovation that helped drive the rapid diversification of terrestrial arthropod communities during the Devonian period. The water cycles in the Devonian world would have been fundamentally different from those we experience today, operating on scales and following patterns that have no modern equivalent. The warm, humid climate meant that vast quantities of water were constantly evaporating from the extensive wetlands and shallow seas that covered much of the planet's surface. This water vapour would have been carried by atmospheric circulation patterns to distant regions, where it would have condensed and fallen as precipitation. But the distribution of this precipitation would have been much more uniform than in our modern world. Without the sharp temperature gradients that drive modern weather systems, without the dramatic topographic barriers created by major mountain ranges, the Devonian atmosphere would have been characterized by gentler, more predictable circulation patterns that distributed moisture relatively evenly across the planet's surface. This meant that the fungal towers would have experienced remarkably stable hydrological conditions. Unlike modern plants, which must cope with seasonal droughts and irregular precipitation patterns, the giants could rely on consistent moisture availability throughout their long lifespans. This hydrological stability was probably crucial for their success, allowing them to grow to enormous sizes without the sophisticated water conservation and transport systems that characterize modern trees. The interaction between the hydrological cycle and the vast fungal biomass would have created feedback effects that stabilized the climate system. The towers themselves would have been major sources of water vapor, releasing moisture through their surfaces and creating local humidity that promoted continued fungal growth. This biological humidity pump would have helped maintain the moist conditions that the fungi required, creating a self-reinforcing system that promoted ecosystem stability. The rivers and streams of the Devonian world would have been very different from modern waterways. Without the complex root systems of trees to stabilize riverbanks, without the sophisticated erosion control provided by modern plant communities, Devonian rivers would have been more dynamic and unstable, frequently changing course and creating the kind of constantly shifting wetland environments that the fungal giants preferred. These dynamic waterways would have been important highways for the dispersal of fungal spores and other microscopic life forms. River systems would have connected distant fungal populations, allowing genetic exchange across vast distances and preventing the isolation that might have led to local extinctions. The constant movement of water would have created a planetary circulation system for microscopic life, mixing and redistributing organisms across entire continents. The chemistry of Devonian waterways would have been distinctive as well. The high atmospheric carbon dioxide levels would have made rainwater more acidic than today, accelerating the weathering of rocks and increasing the concentration of dissolved minerals in surface waters. This enhanced mineral availability would have supported more productive aquatic ecosystems and provided essential nutrients for the terrestrial organisms that depended on these water sources. The estuarine environments where rivers met the sea would have been particularly important for the fungal giants. These transitional zones would have provided optimal conditions for large-scale decomposition, with abundant organic matter from both terrestrial and marine sources, constant moisture, and the chemical gradients that fungi need for efficient nutrient processing. Many of the largest prototaxoids fossils are found in rocks that were deposited in these estuarine environments. The tidal rhythms in these coastal areas would have created twice-daily cycles of inundation and exposure that would have been important environmental drivers for many organisms. The fungal towers that lived in these areas would have been adapted to these tidal cycles, perhaps timing their growth and reproductive activities to take advantage of the nutrients and moisture that came with each high tide. The magnetic field of the Earth during the Devonian period was different from today's, both in its strength and in its orientation. The magnetic poles were located in different positions relative to the geographic poles, and the field itself may have been weaker or more variable than our current magnetic field. These differences would have affected the atmospheric chemistry and the radiation environment that organisms experienced. A weaker magnetic field would have allowed more cosmic radiation to reach the Earth's surface, potentially affecting mutation rates and evolutionary processes. This increased radiation exposure might have contributed to the rapid evolution and diversification that characterized life during the Devonian period. It might also have influenced the atmospheric chemistry, promoting the formation of compounds that don't exist in significant quantities under our current magnetic conditions. The different orientation of the magnetic field would have affected atmospheric circulation patterns, potentially contributing to the more uniform climate that characterized the Devonian world. Without the magnetic field configurations that help drive modern jet streams and weather systems, atmospheric circulation would have followed different patterns, creating the stable, predictable climate conditions that allowed the fungal giants to thrive. These magnetic field differences would also have affected any organisms that use magnetic navigation, though we don't have clear evidence of whether any Devonian organisms had evolved magnetic sensory capabilities. Modern research shows that many organisms, from bacteria to birds, use magnetic fields for navigation and orientation. If similar capabilities existed in Devonian organisms, the different magnetic field configuration would have created very different navigational landscapes. The study of ancient magnetic fields, called paleomagnetism, provides important constraints on our understanding of Devonian climate and atmospheric conditions. By analysing the magnetic signatures preserved in ancient rocks, scientists can reconstruct the orientation and strength of ancient magnetic fields, providing clues about the environmental conditions that existed when those rocks were formed. The soil development processes in the Devonian world were unlike anything we see in modern terrestrial environments. Without the complex root systems and leaf litter inputs that characterize modern forest soils, Devonian soils developed through different processes and had very different properties from modern soils. The primary soil-forming process was the weathering of bedrock by the slightly acidic rainwater that fell in the high carbon dioxide atmosphere. This chemical weathering proceeded more rapidly than in our modern world, breaking down rocks and creating the mineral substrates that supported early terrestrial life. But without plant roots to physically break apart rocks and create the complex soil structures we're familiar with, soil development was largely a chemical rather than a biological process. The organic matter in Devonian soils came primarily from the remains of primitive plants, algae, and microorganisms, rather than from the complex leaf litter and root systems that dominate modern soil organic matter. This created soils with different chemical and physical properties, soils that were probably less structured and less chemically complex than modern forest soils. The fungal giants would have been major contributors to soil development, both through their direct decomposition activities and through the organic matter they added to the soil when they died. The massive amounts of fungal biomass that accumulated and decomposed in Devonian soils would have created organic-rich layers that had no equivalent in modern terrestrial environments. These fungal-rich soils would have had unique chemical properties, rich in the complex organic compounds that fungi produce during their metabolic processes. Some of these compounds would have acted as natural preservatives, helping to stabilize organic matter and prevent rapid decomposition. Others would have served as chemical signals, facilitating communication between different organisms in the soil community. The absence of earthworms and other soil-mixing organisms that are common in modern soils meant that Devonian soils would have been more stratified, with distinct layers that reflected different periods of deposition and decomposition. These layered soils would have created diverse microhabitats for smaller organisms with different chemical and physical conditions at different depths. The nitrogen cycle in Devonian soils would have operated very differently from modern nitrogen cycles. Without the leguminous plants that fix atmospheric nitrogen in modern ecosystems, the primary source of biologically available nitrogen would have been lightning and the activities of nitrogen-fixing bacteria. This might have made nitrogen a limiting nutrient in many Devonian ecosystems, potentially constraining the growth and productivity of early terrestrial life. The phosphorus cycle would also have been different, with fewer biological mechanisms for extracting phosphorus from rocks and making it available to living organisms. The fungal giants would have been important players in the phosphorus cycle, using their powerful biochemical systems to extract phosphorus from organic matter and mineral sources and making it available to other organisms through their decomposition activities. Understanding these ancient soil systems has important implications for modern agriculture and soil management. The principles that governed nutrient cycling in Devonian soils, the role of fungi in soil development, the importance of organic matter stabilization, all of these factors continue to influence modern soil systems and could inform more sustainable approaches to soil management and agricultural production. The atmospheric pressure during the Devonian period was probably somewhat different from today's, though the exact magnitude of this difference is difficult to determine from the geological record. Changes in atmospheric pressure would have affected many biological processes, from respiration and circulation to the physics of spore and pollen dispersal. Higher atmospheric pressure would have made the air denser, potentially improving the efficiency of gas exchange for terrestrial organisms, but also making it more difficult for spores and other small particles to remain airborne for long-distance dispersal. Lower pressure would have had the opposite effects, making respiration more challenging but facilitating long-distance dispersal of reproductive propagules. The density of the atmosphere would also have affected the structural requirements for large terrestrial organisms. In a denser atmosphere, the increased air resistance would have provided more support for tall structures, potentially making it easier for the fungal giants to achieve their remarkable heights. In a less dense atmosphere, structural support would have been more challenging, requiring stronger and more sophisticated support systems. The acoustic properties of the Devonian atmosphere would have been different as well, affecting any sound-based communication that might have existed among early terrestrial organisms. Different atmospheric composition and pressure would have changed the speed of sound transmission and the attenuation of acoustic signals, creating different acoustic landscapes for any organisms that relied on sound for communication or navigation. While we don't have clear evidence that any Devonian organisms used sophisticated acoustic communication, the physical properties of the atmosphere would have constrained the evolution of such systems. Understanding these acoustic constraints helps us understand why certain types of communication systems did or didn't evolve during different periods of Earth's history. The study of ancient atmospheric conditions requires sophisticated modelling and analysis of multiple types of geological evidence. Scientists use ice core data, analysis of ancient atmospheres trapped in rocks, isotopic analysis of ancient organic matter, and computer models of ancient climate systems to reconstruct the atmospheric conditions that existed during different periods of Earth's history. These reconstructions are constantly being refined as new analytical techniques become available, and as our understanding of the relationships between atmospheric conditions and geological processes improves. Each new piece of evidence helps build a more complete picture of what the Devonian world was like and how it differed from our modern environment. The implications of this atmospheric research extend beyond paleontology into fields like climate science and astrobiology. Understanding how different atmospheric conditions affect the evolution and distribution of life helps us understand how life might respond to future changes in Earth's atmosphere, and how life might evolve on other planets with different atmospheric compositions. The extinction events that mark the end of the Devonian period were complex and protracted, involving multiple episodes of environmental change that gradually transformed terrestrial ecosystems over millions of years. These extinctions weren't sudden catastrophes, but rather slow-motion ecological transformations that reshaped the biological world. The late Devonian extinctions affected both marine and terrestrial life, but their impacts on land were particularly profound. The fungal giants were among the casualties, gradually declining in abundance and diversity as environmental conditions shifted and new forms of life began to dominate terrestrial ecosystems. The causes of these extinctions are still debated among scientists, but they probably involved a combination of climate change, ocean chemistry changes, and the evolution of new forms of life that altered competitive relationships in terrestrial ecosystems. The rise of the first trees created new forms of ecological competition that the fungal giants were ill-equipped to handle. The cooling climate that characterized the end of the Devonian period would have been particularly challenging for organisms that had evolved in the warm, stable conditions of the mid-Devonian greenhouse world. The fungal giants, with their large size and slow growth rates, would have been especially vulnerable to rapid environmental changes. But the extinctions that ended the age of mushroom rulers also created opportunities for new forms of life to evolve and diversify. The ecological niches that had been occupied by the fungal giants became available for colonization by early trees and other plant groups, setting the stage for the evolution of the complex forest ecosystems that would characterize the Carboniferous period. The transition from the Devonian to the Carboniferous represents one of the most profound ecological transformations in Earth's history. It marked the end of ecosystems dominated by decomposer organisms and the beginning of ecosystems dominated by primary producers. This shift had cascading effects throughout the biosphere, influencing everything from global climate patterns to the evolution of animal life. Understanding these ancient extinction events has important implications for modern conservation biology and ecosystem management. The patterns of extinction and recovery that characterize the end of the Devonian period provide insights into how ecosystems respond to environmental change and how long recovery processes take after major ecological disruptions. The study of these ancient extinctions also provides perspective on the current biodiversity crisis. While the scale and pace of modern extinctions are unprecedented in many ways, the ecological principles that governed ancient extinction and recovery processes continue to operate in modern ecosystems. Understanding these principles helps us predict how current extinction processes might unfold and how we might intervene to prevent or mitigate their effects. As we conclude our journey through the world of mushroom rulers, it's worth reflecting on what these ancient giants can teach us about resilience, adaptation, and the long-term perspectives that are essential for understanding life on Earth. The fungal networks that descended from the Devonian giants continue to operate according to principles that have been tested over hundreds of millions of years of evolutionary time. These principles, cooperation over competition, long-term stability over short-term gain, network effects over individual achievement, represent time-tested strategies for creating resilient and sustainable biological systems. They offer alternatives to the more competitive and individualistic approaches that often dominate modern human societies. The perspective that comes from studying deep time, from understanding the vast scales over which life operates and evolves, can be both humbling and inspiring. It reminds us that our individual lives, our civilizations, our species, are all part of larger patterns that extend far beyond our immediate experience or understanding. But it also connects us to something larger and more enduring than ourselves. The networks that supported the Devonian giants are still operating beneath our feet, still processing information, still making decisions, still supporting the biological world that we depend on for our survival. We're not separate from these ancient systems, we're part of them, embedded in networks of relationship and dependency that connect us to the deepest history of life on Earth. There's something profoundly comforting about understanding these connections, about recognizing that we're supported by systems that have been operating successfully for hundreds of millions of years. The challenges we face as a species, environmental degradation, climate change, social inequality, are serious and urgent, but they're not unprecedented. Life has faced and survived many challenges over its long history, often emerging stronger and more diverse than before. The fungi that once ruled the Earth as towering giants now rule as invisible networks, connecting and supporting the biological world in ways that are less visible, but perhaps more important than their ancient architectural achievements. They remind us that success can take many forms, that adaptation often involves fundamental changes in strategy and approach, and that the most important work is often done quietly, behind the scenes, by systems that most people never notice or think about. As you drift towards sleep tonight, you can carry with you the understanding that you're part of these ancient stories, connected through invisible networks to forms of life that have been experimenting with cooperation and mutual support for hundreds of millions of years. The wisdom of the mushroom rulers, their understanding of long-term thinking and network effects, their sophisticated approaches to information processing and resource sharing continue to operate in the world around you. The age of mushroom rulers ended long ago, but their legacy lives on in every forest, every garden, every handful of soil. Their descendants continue to grow, to connect, to support the magnificent diversity of life that surrounds us. And in their quiet, persistent way, they remind us that cooperation, patience, and long-term thinking might be the most important qualities we can cultivate as we face the challenges and opportunities of our uncertain future. Sweet dreams from all of us here at Uniscope. Until next time, keep wondering about the hidden connections that make life possible, the ancient partnerships that support our modern world, and the quiet experiments in cooperation that continue all around us, mostly unseen but absolutely essential. The networks that sustain the mushroom rulers are still here, still working, still growing, connecting us all in the vast web of life that spans deep time and continues into whatever future awaits us on this ancient, ever-changing, endlessly fascinating planet we call home.